This lecture concerns an overview of body systems. Life is characterized by a hierarchy of organization in animals. Individual cells are grouped into tissues, which are in turn grouped into organs, which participate in organ systems, which together make up the entire organism. Parts of the body at each level of the hierarchy act together to perform the functions of life, such as regulating an animal's internal environment. For example, the regulation of body temperature within an athlete's body requires cooperation of several levels of body organization. The brain, an organ, sends the signals via the nervous system that trigger specific changes in the tissues and individual cells. The result is a change within the whole body. But what is a better tool, a hammer or a screwdriver? The answer, of course, depends on what you're trying to do. Given a choice of tools, you would not use a hammer to loosen a screw or a screwdriver to pound a nail. How a device works is correlated with its structure. Form fits function. A chair, for example, must have a surface to sit on and a stable base to support that surface. These structural elements are dictated by the task that the chair must perform. The same principle applies to life at its many structural levels, from cells to organisms. Analyzing a biological structure gives us clues about what it is and how it works. Conversely, knowing the function of a structure provides insight about its construction. In exploring life at its many levels, we will discover functional elegance at every turn. When discussing form and function, biologists distinguish anatomy from physiology. Anatomy is the study of the structure or form of an organism and its parts. Physiology, on the other hand, is the study of the function of an organism's structural equipment. For example, an anatomist might study the arrangement of blood vessels near the surface of the skin, whereas a physiologist might study how those blood vessels dilate in response to a sudden rise in temperature. Despite their different approaches, the two disciplines serve the same purpose: to better understand the connections between form and function. The correlation of form and function is an important fundamental principle of biology that is evident at all levels of life's hierarchy. It is a principle that will guide us throughout our study of animals. But this rule of quote-unquote design does not mean that such biological tools as a bird's wings, a fish's gills, or a mammal's teeth are products of purposeful invention. Here, the analogy to household tools such as hammers and screwdrivers fails because those tools were designed with specific goals. It is natural selection that refines biological equipment. It does so by screening for the most effective variations among individuals of a population, those variations that are most advantageous in the local environment. Generations of selecting for what works best in a particular environment will fit form to function without any goal-oriented plan. Thus, the form-function principle is just another facet of biology's unifying theme, that is, evolution. So recall from a previous chapter that the cell is the basic unit of all living organisms. In most multicellular animals, including humans, cells usually do not act alone, but instead are grouped into tissues. A tissue is an integrated group of similar cells that performs a specific function. The cells composing a tissue are specialized. They have an overall structure that enables them to perform a specific task. Some tissues consist of living cells surrounded by a meshwork of non-living fibers. The cells of other tissues are held together by a sticky substance that coats the cells or by special junctions that rivet cells together. An animal has four main categories of tissue: epithelial, connective, muscular, and nervous. And the structure of each type of tissue relates to its specific function. The next level in the structural hierarchy is the organ. An organ consists of two or more tissues packaged into one working unit that performs a specific function. Your heart, brain, and small intestine are examples of organs. At each level of the hierarchy, functions emerge from the collective interaction of the structures at lower levels. An organ performs functions that none of its component tissues can carry out alone. To see how multiple tissues coordinate a single organ, examine the layered arrangement of tissues in the wall of the small intestine. The inside wall is lined with epithelial tissue, that is the top layer of cells that secretes mucus and digestive enzymes. Underneath this layer are 2 layers of connective tissue, one containing blood capillaries and lymph vessels separated by 2 layers of smooth muscle. A final layer of epithelial tissue forms the outside surface. Neurons regulate the rhythmic contractions of the intestinal muscles that propel food along the digestive tract. The organs of the human and most other animals make up organ systems, that is, teams of organs that work together to perform vital body functions. The components of an organ system can be either physically connected or dispersed within the body. An example of an organ system is your circulatory system, which transports materials throughout your body. Its main organs are the heart and blood vessels. An organism depends on the coordination of all its organ systems for survival. For instance, nutrients that are absorbed from the digestive tract are distributed throughout the body by the circulatory system. But the heart that pumps the blood through the circulatory system requires nutrients that are absorbed from food by the digestive tract, and it also requires oxygen that is obtained from the air by the respiratory system. The failure of any organ system will jeopardize the entire animal because organ systems are so interwoven and dependent upon one another. Your body is a whole, living unit that is greater than the sum of its parts. And on this next slide, we just see a table that describes the structure and function of organs. So I'll give you a few moments to go through this table on your own time. And on this slide, we see a table that outlines organ systems. Take a few minutes of your own time to go through this table. And in this slide, we see a pictorial representation of the 11 different organ systems. In the upcoming slides, we will go through each of these organ systems one by one. But before we do that, let's talk about homeostasis. So most animals are covered with a protective layer that separates the external world from the internal chemical environment. This does not mean, however, that an animal barricades itself against the world outside. On the contrary, every organism is an open system, meaning that it continuously exchanges chemicals and energy with its surroundings. In fact, organisms must do so to survive. You eat, breathe, defecate, urinate, sweat, and radiate heat. These are all examples of how you operate as an open system. And the exchange of materials extends downward to the cellular level. Nutrients and oxygen must enter every living cell, and carbon dioxide and other wastes must exit. An animal's size and shape affect how it exchanges energy and materials with its surrounding environment. In an animal body, every living cell must be bathed in a watery solution, partly because substances must be dissolved in water to cross cell membranes. An amoeba, which is a type of a protist, lives in water and consists of only one cell. Thus, no part of its body is very far away from its outer surface where the exchange with its watery environment can occur. Exchange with the environment is more complicated for multicellular organisms. Each microscopic cell in a multicellular organism has a plasma membrane where the exchange of materials can occur. But this exchange only works if all cells of the animal have access to a suitable watery environment. A hydra, which is a simple pond-dwelling relative of jellyfishes, has a body wall only 2 cell layers thick. Both the outer layer and inner layer of cells are bathed in pond water, which enters the digestive sac through the mouth. Every cell of the hydra can thus exchange materials through direct contact with the watery environment. A flat body shape is another way of maximizing exposure to the environment. For instance, a tapeworm may be several meters long, but because it is very thin, most of its cells are bathed in the body fluid of the worm's host, from which it obtains its nutrients. Simple body forms, such as two-layered sacs and flat shapes, do not allow for much complexity in internal organization, but animals with more complex body forms face the same basic problem. Every living cell in the body must be bathed in fluid and have access to oxygen, nutrients, and other resources from the outside environment. Complex animals have extensively folded or branched internal surfaces that maximize the area for exchange with the immediate environment. For example, your lungs, which exchange oxygen and carbon dioxide with the air you breathe, are not shaped like big balloons, but like millions of tiny balloons at the tips of finely branched air tubes. The epithelium of the lungs has a very large total surface area, about the size of a tennis court. Every living organism has the ability to respond to its environment. Some of these responses prevent changes in the outside world from adversely affecting the internal makeup of the organism. In this section, we're going to learn about how animals adjust to the changing environmental conditions, looking in particular at how they regulate temperature and control the movement of water and solutes. One of the body's most important functions is to maintain its integrity even when the world around it changes. The internal environment of vertebrates consists of interstitial fluid that fills spaces between cells and exchanges nutrients and wastes with microscopic blood vessels. It is important that the composition of the interstitial fluid remains relatively constant no matter what occurs in the outside world. Homeostasis, which literally means steady state, is the tendency to maintain relatively constant conditions in the internal environment even when the external environment changes. In the face of large external changes, the mechanisms of homeostasis normally maintain internal conditions within a range that the animal's metabolism can tolerate. For example, your internal body temperature normally fluctuates by less than a degree regardless of the temperature of your surroundings. Actually, the internal environment of an animal always fluctuates slightly in response to internal and external changes. Homeostasis is a dynamic state and interplay between the outside forces that tend to change the internal environment and the internal control mechanisms that oppose such changes. Changes do occur, but they are generally moderated to stay within a range that is tolerable for all living cells. Additionally, there are times during the development of an animal when major changes in the internal environment are pre-programmed to occur. For example, the balance of hormones in human blood changes radically during puberty or pregnancy, and the internal temperature may rise in response to a bacterial infection. Most mechanisms of homeostasis depend on a principle called negative feedback. That is a form of regulation in which the results of a process inhibit that same process. The concept of negative feedback can be outlined with household homeostasis, that is the control of room temperature. During the winter, a home heating system maintains a relatively constant temperature inside the house despite drastic changes that may occur outside. A key component of this is a control center, that is a thermostat, that monitors the temperature and switches the heater on and off. Whenever room temperature drops below the set point, the thermostat switches the heater on. When the temperature rises above that set point, the thermostat switches the heater off. This homeostatic mechanism is called thermoregulation. As you walk from one warm building to the next on a cold winter day, your body's temperature will barely fluctuate despite the drastic change in temperature in the environment around you. The ability to maintain a body temperature substantially warmer than the surrounding environment is a characteristic of endotherms. Endotherms are animals, such as mammals and birds, that derive most of their body heat from their metabolism. In contrast, ectotherms, which include most invertebrates, fishes, amphibians, and non-bird reptiles, obtain their body heat primarily by absorbing it from their surroundings. Humans have a number of structures and mechanisms that aid in thermal regulation. Our brain has a control center that maintains our body temperature that is nearly 37 degrees Celsius or 98.6 degrees Fahrenheit. When body temperature falls below normal, as in outside frigid weather, for example, the brain's control center sends signals that trigger changes that will bring it back to normal. Blood vessels near the body's surface constrict, this conserves heat, and muscles contract, causing us to shiver, this produces heat. When body temperature gets too high, the control center sends signals to dilate blood vessels near the skin and activate sweat glands, and this allows excess heat to escape. Like a home controlled by a thermostat, the temperature of the body does not stay completely constant but fluctuates up and down within an acceptable range. Fever is an abnormally high internal temperature. This is a body-wide response that usually indicates an ongoing fight against infection. When the immune system cells encounter invading microbes, the cells release chemicals that travel through the bloodstream to the brain's thermostatic control center. These chemicals stimulate the control center to raise the body's internal temperature, producing a fever. Many people mistakenly believe that invading microbes themselves cause a fever. In fact, the cause is usually the body's fight against the microbes. A fever over 40 degrees Celsius or 100 degrees Fahrenheit may be life-threatening because a temperature that high can damage body proteins. A moderate fever of, say, 39 to 38 degrees Celsius, which is roughly 100 to 102 degrees Fahrenheit, however, discourages bacterial growth and speeds the body's internal defenses. A moderate fever thus helps protect the body's internal environment against potentially harmful invaders from the external environment. Moving on, calcium ions are used for the heartbeat, the contraction of muscles, the activation of enzymes, and cellular communication. The parathyroid and thyroid glands of the endocrine system detect changes in blood calcium levels. When the parathyroid glands detect low blood calcium levels, the skeletal, urinary, and digestive systems all act as effectors to achieve this goal through the negative feedback loop. Moving on, the endocrine functions of the pancreas and liver coordinate efforts to maintain normal blood glucose levels. The nervous and digestive systems also play a role in maintaining blood glucose levels by prompting you to eat, which raises your blood glucose level. This is another example of a homeostatic mechanism. So let's begin our discussion of the various organ systems of the body, and we'll begin with the nervous system. The nervous system forms a communication and coordination network throughout an animal's body. Nervous systems are the most intricately organized data processing systems on Earth. Your brain, for instance, contains an estimated 100 billion neurons. Those are nerve cells specialized for carrying electrical signals from one part of the body to another. Each neuron may communicate with thousands of others, forming networks that enable us to move, perceive our surroundings, learn, and remember. The nervous system of most animals have two main anatomical divisions: the central nervous system, or the CNS, which consists of the brain and invertebrates, the spinal cord, and the peripheral nervous system, or the PNS, which is made mostly of nerves that carry signals into and out of the CNS. A nerve is a communication line made from cable-like bundles of neuron fibers tightly wrapped in connective tissue. There are three types of neurons that carry out the functions of the nervous system. Sensory input is the process of sending signals from sensory receptors, such as light-detecting receptors in the eyes, to the central nervous system. Sensory neurons convey this information. Integration is the interpretation of sensory signals and the formulation of responses. Interneurons are located entirely within the central nervous system, and they perform integration. Motor output is the process of sending signals from the integration centers to the effectors, such as muscle cells. Effectors perform the body's responses, that is, moving a leg, for example. Motor output is performed by motor neurons. So to summarize, you've got sensory neurons, interneurons, and motor neurons, and those are the three types of neurons in your nervous system. And in this next slide, we see a pictorial representation of both the central nervous system and the peripheral nervous system. Moving on, the human endocrine system consists of about a dozen major glands. Some of these, such as the thyroid and pituitary gland, are endocrine specialists. That is, their primary function is to secrete hormones into the blood. Several other glands have both endocrine and non-endocrine functions. The pancreas, for example, secretes hormones that influence the level of blood glucose in the blood and also secretes digestive enzymes into the intestine. Other organs are primarily non-endocrine but have some cells that secrete hormones. For example, the stomach is primarily a digestive organ but also releases a hormone that travels to the brain and stimulates appetite. And the cartoon in this slide shows the locations of the major endocrine glands of the human body. This cartoon provides an overview of the endocrine system. Hormones have a wide range of targets. Some hormones, like the sex hormones, affect the tissues of the body. Others, such as glucagon from the pancreas, have only a few kinds of target cells, in this case the liver and fat cells. In some cases, a hormone elicits different responses in different target cells depending on the type of cell and its signal transduction pathway. And in this slide, we see a table of some examples of hormones. So I'll give you a few moments to go through this slide on your own time. And on this slide, it's just a continuation of the previous table. Please review this table at your own leisure. I always thought that the reproductive system was very interesting in the respect that it is the one system that you can live without. Life cannot continue without your cardiovascular system, your respiratory system, your skeletal system. If you lose those systems, you can't survive. But without the reproductive system, you can still continue to survive. You may not be able to reproduce and continue the species, but you yourself could maintain homeostasis and continue with your life. So let's have a closer look at our reproductive system. Although every individual animal has a relatively short lifespan, species last much longer because of reproduction, that is, the creation of new individuals from existing ones. Animals reproduce in a great variety of ways, but there are two principal modes: asexual reproduction and sexual reproduction. Asexual reproduction is the creation of offspring that are genetically identical to the lone parent. Several types of asexual reproduction can be found among animals. Many invertebrates reproduce asexually by budding, the outgrowth and eventual splitting off of a new individual from a parent. Hydras, relatives of jellyfishes, reproduce this way. The sea anemone also undergoes fission, which is the separation of a parent into two or more offspring of about equal size. Another type of asexual reproduction involves fragmentation, the breaking of the body into several pieces, followed by regeneration, the regrowth of lost body parts. Some organisms have remarkable powers of regeneration. In certain species of sea stars, for example, an arm that is split off can give rise to a new sea star. In the case of some species of sea sponges, if a single sponge is pushed through a wire mesh, each of the resulting clumps of cells can regrow into a new sponge. In nature, asexual reproduction has several potential advantages. Because it eliminates the need to find a mate, asexual reproduction allows the individual members of a species to perpetuate themselves even if they are isolated from one another. Asexual reproduction also allows organisms to multiply quickly without spending time or energy producing sperm and eggs. If an individual is very well suited to its environment, asexual reproduction allows it to reproduce rapidly and exploit available resources. A potential disadvantage of asexual reproduction is that it produces genetically uniform populations. Genetically similar individuals may thrive in a particular environment, but if the environment changes and becomes less favorable to survival, all individuals may be affected equally and the entire population may die out. On the other hand, there's sexual reproduction. You know that you have a mix of traits from your mother and father, but you also have a genome distinct from every other human unless you're an identical twin. You are the product of sexual reproduction, the creation of offspring by the fusion of two haploid sex cells called gametes to form a diploid zygote. The male gamete is the sperm and the female gamete is the egg. The zygote and the new individual it develops into contain a unique combination of genes carried from the parents via the egg and sperm. Unlike asexual reproduction, sexual reproduction increases genetic variability among offspring as a result of the huge variety of gametes produced by meiosis. Variation is the raw material of evolution by natural selection. In theory, when an environment changes, there is a better chance that some of the variant offspring will survive and reproduce than if all offspring are genetically very similar. Some animals can reproduce both sexually and asexually, benefiting from both modes. An example of this is the organism called a starlet sea anemone. Why would such dual reproductive capabilities be advantageous to an animal? Some animals reproduce asexually when food is ample and conditions are favorable. But when conditions change, that is, they become colder or drier, for example, these animals switch to sexual reproduction, producing a new generation of genetically varied individuals with a better potential to adapt to the changing conditions. In some species, each individual is a hermaphrodite, meaning that it has both male and female reproductive systems. When earthworms mate, for example, each individual serves as both the male and the female, donating and receiving sperm, but most animals are of a single sex. The mechanics of fertilization play an important part in sexual reproduction. Many aquatic animals use external fertilization, which the parents discharge their gametes into the water where fertilization occurs. The female and male don't necessarily have to touch to mate. In contrast, nearly all terrestrial animals reproduce when sperm are deposited by a male within the female's body. Although the differences between human male and human female reproductive anatomies are obvious, there are also similarities. Both sexes have a pair of gonads, the organs that produce gametes, and both sexes have ducts that store and deliver the gametes as well as structures that allow mating. We'll now explore the anatomical differences of the human male and the human female. In this slide, this cartoon shows the male reproductive system. The penis contains erectile tissue that can fill with blood and cause an erection during sexual arousal. The penis consists of a shaft that supports a highly sensitive glands or a head. A prepuce, or foreskin, covers the glands. The foreskin may be surgically removed in a procedure known as circumcision. The testes, the male gonads, are located outside of the abdominal cavity in a sac called the scrotum. Sperm cannot develop at body temperature. By keeping the sperm-forming cells away from the body, the scrotum keeps them cool enough to develop normally. In cold conditions, muscles around the scrotum contract, pulling the testes towards the body, maintaining the proper temperature. At the onset of puberty, to the death of the male, the testes produce hundreds of millions of sperm every day. From the testes, the sperm pass into a coiled tube called the epididymis. During ejaculation, that is the expulsion of sperm containing fluid from the penis, the sperm leave the epididymis and travel through a duct called the vas deferens. The seminal vesicles and prostate gland add fluid that nourishes the sperm and provides protection from the natural acidity of the vagina. The prostate gland is the source of some of the most common medical problems in men over 40. Prostate cancer, for example, is the second most commonly diagnosed cancer in the United States after skin cancer. Each vas deferens, one from each of the two testes, empties into the urethra. The urethra conveys, at different times, both sperm and urine out through the penis. Ejaculation, which is caused by the contraction of muscles along the sperm ducts, releases about 5 milliliters or 1 teaspoonful of semen. Only 5% of semen consists of sperm, typically 200 to 500 million of them. The remaining 95% of semen is fluid secreted by the various glands. And in this slide, we see a table outlining the components of the male reproductive system. Please spend a little bit of your own time reviewing this table. And in this slide, we see two views of the female reproductive system in cartoon format. The outer features of the female reproductive anatomy are collectively called the vulva. The vagina, or birth canal, opens to the outside just behind the opening of the urethra, the tube in which urine is excreted. An outer pair of thick, fatty ridges, called the labia majora, protects the entire genital region, and a pair of inner skin folds called the labia minora borders the openings. A thin membrane called the hymen partly covers the vaginal opening until sexual intercourse or other vigorous physical activity ruptures it. The clitoris is an organ that engorges with blood and becomes erect during sexual arousal. It consists of a shaft supporting a round glans or head covered by a small hood of skin called the prepuce. The clitoris has an enormous number of nerve endings and is very sensitive to touch. The ovaries are the site of gamete production, the gonads, in human females. A woman's ovaries are each about an inch long and have a bumpy surface. The bumps are follicles, each consisting of a single developing egg cell surrounded by one or more layers of cells that nourish and protect it. The follicles also produce estrogen, which is the female sex hormone. A female human is born with over 1 million follicles, but only several hundred will release egg cells during her reproductive years. Starting at puberty and continuing until menopause, that is the end of fertility, which usually occurs during middle age, one follicle, or rarely two or more, matures and releases its egg cell about every 28 days. An egg cell is ejected from the follicle in the process called ovulation. After ovulation, what remains of the follicle grows to form a solid mass called the corpus luteum, which plays a crucial role in the release of hormones during the reproductive cycle. The released egg enters an oviduct, which is also known as a fallopian tube, where cilia sweep it towards the uterus. If sperm are present, fertilization may take place in the upper part of the oviduct. If the released egg is not fertilized, it is shed during menstruation and a new follicle matures during the next cycle. The uterus, which is also called the womb, is the actual site of pregnancy. The uterus is about the size and shape of an upside-down pear, but can grow to several times that size to accommodate a growing fetus. The uterus has a thick, muscular wall lined with a blood-rich layer of tissue called the endometrium. An embryo implants in the endometrium and grows there. The term embryo is used for the stage in development from the first division of the zygote until the body structures begin to appear at about the 9th week of pregnancy. From the 9th week until birth, a developing human is called a fetus. The narrow neck at the bottom of the uterus is called the cervix. It is recommended that a woman have a yearly Pap test in which cells are removed from around the cervix and examined under a microscope for signs of cervical cancer. Regular Pap smears greatly increase the chances of detecting cervical cancer early and therefore treating it successfully. The cervix opens into the vagina. During sexual intercourse, the vagina receives the penis and serves as a repository for sperm. And in this slide, we see a table outlining the components of the female reproductive system. Please spend a little bit of your own time reviewing this table. And in this table, we compare and contrast similar structures in the human reproductive organs. Moving on, in this section we'll focus on the human body's sensory structures. Sensory input is the process of using receptors to sense the environment and send information about it to the central nervous system that will be integrated and acted upon. Sensory receptors, such as the sensory cells in your eyes and the taste buds of your tongue, are tuned to the condition of both the external world and the internal organs. Sensory receptors detect stimuli such as chemicals, light, sounds, cold, heat, and touch. The sensory receptors in your eyes, for instance, detect light energy. Those in your taste buds detect chemicals dissolved in saliva, and the sensory receptor's job is completed when it sends information to the CNS by triggering action potentials. But what exactly do we mean when we say a sensory receptor detects a stimulus? In stimulus detection, the receptor cell converts one type of signal, the stimulus, to an electrical signal. The conversion is called sensory transduction, and this occurs as a change in the membrane potential of the receptor cell. And in this slide, we outline the sensory system. We see the exteroceptors, vision, hearing, touch, equilibrium, chemical reception. And then we see the interoceptors, which is blood pressure and changes in glucose and pH levels. And that brings us to the olfactory system. Olfaction is the sense of smell. In human, the sense of smell is received in the nasopharynx. Airborne molecules go into solution on moist epithelial surfaces of the nasal passage. An olfactory receptor neuron sends an impulse via the cranial nerve to the olfactory nerve. And then your brain perceives this as a smell. Moving on, the outer surface of the human eyeball is a tough, whitish layer of connective tissue called the sclera. At the front of the eye, the sclera becomes the transparent cornea. The sclera surrounds a pigmented layer called the choroid. At the front of the eye, the choroid forms the iris, which gives the eye its color. The opening in the center of the iris is called the pupil. Behind the pupil is the disc-like lens, which is held in position by ligaments. At the back of the eyeball is the retina. The retina is just inside the choroid that contains photoreceptor cells. The optic nerve connects the retina with the brain. There are no photoreceptor cells in the part of the retina where the optic nerve passes through the back of the eye. We cannot detect light that is focused on this quote-unquote blind spot, but having two eyes with overlapping fields of view enables us to perceive uninterrupted images. Two fluid-filled chambers make up the bulk of the eye. The large chamber behind the lens is filled with a jelly-like vitreous humor. The much smaller chamber in the front of the lens contains a thinner fluid called the aqueous humor. The humors help maintain the shape of the eyeball. In addition, the aqueous humor supplies nutrients and oxygen to the lens, iris, and cornea and carries off wastes. Blockage of the ducts that drain this fluid can cause glaucoma. This is increased pressure inside the eye that may lead to blindness. If diagnosed early, glaucoma can be treated with various medications that increase the circulation of the aqueous humor. A thin mucous membrane called the conjunctiva helps keep the outside of the eye moist. The conjunctiva lines the inner surface of the eyelids and folds back over the white of the eye, but not the cornea. An infection or allergic reaction may cause inflammation of the conjunctiva, a condition called conjunctivitis or pink eye. Bacterial conjunctivitis usually clears up with antibiotic eye drops. Viral conjunctivitis usually clears up on its own, although it is very contagious, especially among young children. A gland above the eye secretes tears. Tears are a dilute salt solution that is spread across the eyeball by blinking and that drain into the ducts leading to the nasal cavities. This fluid cleanses and moistens the eye surface. Excess secretion in response to eye irritation or strong emotions causes tears to spill over the eyelid and fill the nasal cavities. This will produce a runny nose. The cornea lets light into the eye and also helps focus light. Muscles of the iris regulate the size of the pupil, controlling the amount of light that enters. After going through the pupil, light passes through the lens. The lens focuses light onto the retina by bending light rays. Focusing is accomplished through a change in the shape of the lens. The thicker the lens, the more sharply it bends the light. The shape of the lens is controlled by the muscles attached to the choroid. When the eye focuses on a nearby object, these muscles contract, pulling the choroid layer of the eye inward toward the pupil. This makes the ligaments that suspend the lens slacken. With this reduced tension, the elastic lens becomes thicker and rounder. When the eye focuses on a distant object, the muscles controlling the lens relax, putting tension on the ligaments and flattening the lens. Although the human ear is less sensitive than that of many other animals, it is capable of hearing a great range of auditory signals. The ear is composed of three regions: the outer ear, the middle ear, and the inner ear. The outer ear consists of the flap-like pinna, the bendable structure we commonly refer to as our quote-unquote ear, and the auditory canal. A sheet of tissue called the eardrum separates the outer ear from the middle ear. Both the outer ear and the middle ear are common sites of childhood infections, like swimmer's ear. The middle ear contains three small bones: the hammer or the malleus, the anvil or the incus, and the stirrup or the stapes. The stirrup is connected to the inner ear through an opening in the skull bone. The eustachian tube conducts air between the middle ear and the back of the throat, allowing air pressure to stay equal on either side of the eardrum. This tube is what enables you to move air in or out to equalize pressure, or quote-unquote pop your ears when changing altitude rapidly in an airplane or diving underwater. The inner ear consists of fluid-filled channels in the bones of the skull. One of the channels is called the cochlea, and this is a long coiled tube. Our actual hearing organ, the organ of Corti, is located within the fluid-filled canal inside the cochlea. The organ of Corti consists of an array of hair cells embedded in a structure called the basilar membrane. The hair cells are the receptor cells of the ear. These receptor cells project into the fluid, and the tips of most are in contact with the overlying jelly-like membrane. Sensory neurons at the base of the hair cells connect to the auditory nerve, which connects in turn to the brain. But how does the human ear function? A vibrating object creates sound waves in the surrounding air. The pinna and the auditory canal collect these waves and channel them to the eardrum. The waves make your eardrum vibrate with the same frequency as the sound. Frequency is the number of vibrations per second. From the eardrum, the vibrations are concentrated as they pass through the hammer, anvil, and stirrup in the middle ear. The stirrup transmits the vibrations to the inner ear, producing pressure waves in the fluid within the cochlea. As the pressure waves pass through the cochlea, it makes the basilar membrane vibrate. Vibration of the basilar membrane makes the hair-like projections on the hair cells alternately brush against and draw away from the overlying membrane. When a hair cell's projections are bent, ion channels in its plasma membrane open and positive ions enter the cell. As a result, the hair cell develops a receptor potential and releases neurotransmitter molecules at its synapse with a sensory neuron. In turn, the sensory neuron sends action potentials through the auditory nerve to the brain. How is the volume of a sound determined? The higher the volume of sound, the greater the amplitude of the pressure wave it generates. In the ear, the greater the amplitude, the more vigorous the vibrations of fluid in the cochlea, the more pronounced the bending of the hair cells, and the more action potentials generated in the sensory neurons. And this next slide shows a cartoon cross-section of the cochlea of the ear. Notice the location of the organ of Corti, which is the actual hearing organ of the ear. This organ is located within a fluid-filled canal inside of the spiral of the cochlea. And this is where the receptor cells, or the hair cells, connect to the sensory neurons, which in turn connect it to the auditory nerve in the brain. And this brings us to equilibrium. Equilibrioception, or sense of balance, is one of the physiological senses. It allows humans and animals to walk without falling, and some animals are better at this than humans. All forms of equilibrioception can be described as the detection of acceleration. When the sense of balance is interrupted, it causes dizziness, disorientation, and possibly nausea. Moving on, touch is the first sense developed in the womb and the last sense used before death. With 50 touch receptors for every square centimeter and about 5 million sensory cells overall, the skin is very sensitive, and it is the largest and one of the most complex organs of our body. The touch receptors are grouped by type and include mechanoreceptors that are sensitive to pressure, vibration, and slippage, thermoreceptors, which are sensitive to temperature, and nociceptors, which are responsible for sensing pain. Moving on, every organism must exchange materials and energy with its environment. In simple animals, such as hydras and jellyfishes, nearly all the cells are in direct contact with the outside world. Thus, the cells can easily exchange materials with the environment by diffusion, the spontaneous movement of molecules from an area of higher concentration to an area of lower concentration. However, most animals are too large or too complex for such exchange to take place by diffusion alone. In such animals, a circulatory system facilitates the exchange of materials, providing a rapid, long-distance internal transport system that brings resources close enough to cells for diffusion to occur. Once there, the resources that cells need, such as nutrients and oxygen, enter the cytoplasm through the plasma membrane. Metabolic waste, such as carbon dioxide, diffuse from cells to the circulatory system for disposal. All but the simplest animals have a circulatory system with three main components: a central pump, a vascular system, or a set of tubular vessels, and circulating fluid. The two main types of circulatory systems have evolved in animals. Many invertebrates, including most mollusks and arthropods, have an open circulatory system. The system is called open because the circulating fluid is pumped through open-ended vessels and flows out among the cells. In a closed circulatory system, the circulating fluid is blood, which is confined to the vessels and is distinct from the interstitial fluid, the fluid that fills the spaces around the cells. One or more hearts pump blood into large vessels that branch into smaller ones, coursing through the organs. Earthworms, octopuses, and vertebrates are some examples of animals with a closed circulatory system. The closed circulatory system of humans and other vertebrates, consisting of a heart and blood vessels, is called a cardiovascular system. The heart receives blood in a chamber called the atrium. A second chamber called the ventricle pumps blood away from the heart. Blood is confined in three main types of vessels: arteries, capillaries, and veins. Arteries carry blood away from the heart, branching into smaller arterioles as they approach organs. The blood then flows from the arterioles into capillary beds that are networks of tiny vessels called capillaries that infiltrate nearly every organ and tissue of the body. The thin walls of capillaries allow exchange between the blood and interstitial fluid. In fish, blood is oxygenated in gill capillaries and then travels via arteries to other capillary beds where oxygen enters tissue cells. Capillaries converge into venules, which in turn converge into larger veins that return blood back to the heart. Capillaries are the functional center of the circulatory system. This is where the materials are transferred to and from the surrounding tissues. Arteries and veins primarily transport blood back and forth between the heart and capillaries. And in this slide, we see a cartoon of the human heart. So note here the coronary arteries and the cardiac veins. And in this next slide, we see a cartoon, a close-up of how arteries and veins are connected. So the blood flows from the heart in an artery, and that goes into a smaller branch called an arteriole, and the arterioles move into capillaries. And then the capillaries will connect to the venule, where the deoxygenated blood will then reach a vein and be returned to the heart. Moving on, the circulatory system transports gases and other substances to and from body cells. Also, recall that cellular respiration uses oxygen and glucose to produce water, carbon dioxide, and energy in the form of ATP. All working cells, therefore, require a steady supply of oxygen from the environment and must continuously dispose of carbon dioxide. Earth's main reservoir of oxygen is the atmosphere, which is about 21% oxygen. Terrestrial animals access this oxygen by breathing air. Oceans, lakes, and other bodies of water also contain oxygen in the form of dissolved gas, but it makes up only 3 to 5% of that of the environment. Many aquatic animals obtain oxygen by passing water over their gills. The part of an animal where oxygen from the environment diffuses into living cells and carbon dioxide diffuses out to the surrounding environment is called the respiratory surface. The respiratory surface is usually covered with a single layer of living cells that is thin enough and moist enough to allow rapid diffusion between the body and the environment. The respiratory surface of an animal must be large enough to take up oxygen for every living cell in the body. A variety of respiratory surfaces have evolved. For some animals, such as sponges and flatworms, the plasma membrane of every cell in the body is close enough to the outside of the environment for gases to diffuse in and out. In many animals, however, the bulk of the body does not have direct access to the outside environment, and the respiratory surface is the interface between the environment and the blood, which transports gases to and from the rest of the body. Some animals use their entire outer skin as a respiratory surface. An earthworm, for example, has moist skin and gases that diffuse across its general body surface. This results in an exchange of oxygen and carbon dioxide between the environment and a dense net of capillaries just below the earthworm's skin. For most animals, however, the outer body surface is either impermeable to gases or lacks sufficient area to exchange gases for the whole body. In such animals, specialized regions of the body have extensively folded or branched surfaces that provide a large surface area for gas exchange. Within aquatic environments, the respiratory surfaces are found in extensions of the body surface called gills. Lobsters, which are invertebrates, and fishes, which are vertebrates, are examples of animals with gills. The feathery gills, located on either side of a fish's head, have a respiratory surface area much greater than the rest of the body surface. A fish obtains oxygen by continuously pumping water over its gills. As water passes over the respiratory surface of the gills, gases diffuse between the water and the blood. The blood then carries the oxygen to the rest of the body. Gills, especially those of large active fish, must be very efficient to obtain enough oxygen from water. Indeed, some aquatic animals can remove more than 80% of the oxygen from the water moving over their gills. However, in most land-dwelling animals, the respiratory surfaces are folded into the body rather than extending from it. The infolded surfaces are open to the air only through narrow tubes. These tubes allow the animals to breathe air while retaining the moisture needed to maintain the plasma membranes of the respiratory surface cells. Insects breathe using a tracheal system, that is, an extensive network of branching internal tubes called trachea. Trachea begin near the body's surface and branch down to narrower tubes that extend to every cell. Gas exchange with cells occurs via diffusion across the moist epithelium that lines the tips of the tubes. Because almost every body cell is near the respiratory surface, gas exchange in insects requires no assistance from the circulatory system. Lungs are the most common respiratory system among snails, some spiders, and terrestrial vertebrates, such as amphibians, birds, other reptiles, and mammals. In contrast to trachea that branch throughout an insect's body, lungs are localized organs lined with moist epithelium. And this slide shows a cartoon of a human respiratory system. Moving on, the first step of gas exchange is breathing, the ventilation of the lungs by alternate inhalation and exhalation. When an animal with lungs breathes, a large, moist internal surface is exposed to the air. Oxygen diffuses across the cells lining the lungs and into the surrounding blood vessels. Simultaneously, carbon dioxide passes out of the blood vessels into the lungs and is exhaled. Most of the land animals require a lot of oxygen. The inner tubes of the lungs are extensively branched, providing a large respiratory surface. The second step in gas exchange is the transport of oxygen from the lungs to the rest of the body via the circulatory system. The blood also carries carbon dioxide from the tissues back to the lungs. In the final step of gas exchange, oxygen diffuses from red blood cells into body cells, which release carbon dioxide into the blood. The delivered oxygen is used by the body cells to obtain energy from food via the process of cellular respiration. The same process produces carbon dioxide as a waste product that cells pass to the blood. The circulatory system transports the carbon dioxide back to the lungs where it is exhaled. Moving on, nearly everything in the environment, including most of what we touch or taste, and even the air we breathe, teems with microbes, some of which can make us sick. Yet we do not constantly become ill thanks to the immune system, the body's system of defenses against infectious disease. The human body has 3 cooperative lines of defense that protect us against the constant barrage of pathogens, disease-causing viruses, and microorganisms. The first line of defense is external barriers that prevent pathogens from getting deep inside the body. If that line fails, some internal protections stand ready as a second line of defense. These first 2 lines of defense are innate defenses, that is, they are already present and act the same whether or not an invader has been previously encountered. We call this nonspecific immunity. Nearly all animals have some types of innate defenses. Later, we'll discuss another line of defense called adaptive defenses, or specific immunity. For a foreign substance to get inside the body, it must first get by the body's external barriers. These barriers form an important protective line because they prevent infection as opposed to the body's other defenses, which act only after the infection has occurred. Some barriers block or filter out microbes. Intact skin forms a tough outer layer that most bacteria and viruses cannot penetrate. Inside the nostrils, hairs filter many particles from the incoming air. Organ systems that are open to the external environment, such as the respiratory, digestive, reproductive, and urinary systems, are lined with mucous membranes. These membranes secrete mucus, a sticky fluid that traps bacteria, dust, and other particles. Beating cilia, extending from the cells of the respiratory tract, sweep mucus with the trapped particles outward until it is swallowed or expelled by sneezing, coughing, or blowing the nose. External barriers also include chemical defenses in the form of antimicrobial secretions. Sweat, saliva, and tears contain enzymes that disrupt bacterial cell walls. Glands produce oils and acids that make the skin inhospitable to many microbes. Concentrated stomach acid kills most bacteria that we swallow before they can enter the bloodstream. When an invader breaches the body's external barriers, the problem of how to fend off an attack changes substantially. Once within the body fluids and tissues, the invader is no longer an outsider. To fight pathogens within the body, the animal's immune system must detect foreign particles and cells. In other words, an immune system must carry out recognition, distinguishing non-self from self. When the external barriers fail, a set of internal innate defenses comes into play. The second line of defense consists mainly of white blood cells and defensive proteins. The two types of white blood cells are involved in internal innate defense are phagocytic cells and natural killer cells. Phagocytic cells engulf foreign cells or molecules and debris from dead cells by phagocytosis or cellular eating. Natural killer cells can often recognize virus-infected or cancer cells in the body. When contact is made, natural killer cells release chemicals that kill the diseased cells. The defensive proteins that aid in the innate defense work both indirectly and directly. Interferons produced by virus-infected body cells work indirectly by helping healthy cells resist damage by viral infection. Virus-infected cells make and release interferons that bind to plasma membrane receptors on nearby uninfected cells. This binding stimulates healthy cells to produce proteins that inhibit viral reproduction. Because of their strong antiviral properties, injected interferons are sometimes used to treat certain viral diseases such as hepatitis and genital warts. Complement proteins attack pathogens directly. Some coat the surface of microbes, making them easier for phagocytic cells to engulf. Others cut lethal holes in microbial membranes, causing the invading cells to burst. But what happens if you get a splinter in your finger and don't take care of it? The next day, the finger is often red, swollen, painful, and warm to the touch. These are signs of the inflammatory response to the injury and the infection, another example of an internal innate defense in action. When tissue becomes damaged from cuts, bug bites, or burns, for example, the injured cells and the invading microbes release chemicals that trigger various innate defenses. One such chemical signal is histamine. This causes nearby blood vessels to dilate and leak fluid into the wound tissue, causing it to swell. The swelling helps the tissue to heal by diluting toxins, bringing extra oxygen, and delivering platelets and clotting proteins that promote scabbing. The chemical signal also attracts phagocytic cells. These cells engulf bacteria and the remains of body cells killed by bacteria or by physical injury. The pus that often fills an infected injury consists of phagocytic cells, fluid leaked from capillaries, and other tissue debris. Damaged cells also release chemical signals that increase blood flow to the damaged area, causing the wound to turn red and warm. The word inflammation means setting on fire. In response to severe tissue damage or infection, damaged cells may release other chemicals that travel through the bloodstream to the brain, where they stimulate a fever and may discourage bacterial growth. Anti-inflammatory drugs such as aspirin and ibuprofen dampen the normal inflammatory response and help reduce swelling and fever. This is an example of treating the symptoms of an illness, for example fever, without addressing the underlying cause, which is the injury or infection. All the defenses you've learned about so far are called innate because they're ready, off the rack. That is, innate defenses are present in the body without any preparation. The lymphatic system consists of a branching network of vessels, numerous lymph nodes, and other kinds of organs. The lymphatic vessels carry fluid called lymph, which is similar to interstitial fluid surrounding body cells. The two main functions of the lymphatic system are to return tissue fluid to the circulatory system and to fight infection. When the innate defenses fail to ward off a pathogen, the adaptive defenses, or the specific immunity, kicks in. This line of defense is called adaptive defenses because it is activated only after exposure to specific pathogens. Adaptive defenses depend on white blood cells called lymphocytes that recognize and respond to specific invading pathogens. There are two types of lymphocytes, B cells and T cells. Like all blood cells, they originate from stem cells in the bone marrow. Some immature lymphocytes continue developing in the bone marrow. These become specialized as B cells. Others migrate via the blood to the thymus, a gland in the chest, where they become specialized as T cells. Both B cells and T cells eventually make their way to the lymph nodes and other lymphatic organs. Over 2 trillion lymphocytes are spread throughout the bloodstream and the lymphatic system, making possible an elaborately coordinated response to infection. Whereas the innate defenses are always ready to fight a variety of infections, lymphocytes must be primed by exposure to a foreign molecule. Any molecule that elicits a response from a lymphocyte is called an antigen. Most antigens are molecules on the surfaces of viruses or foreign cells, such as cells of bacteria or parasitic worms. Antigens can also be molecules from mold spores, pollen, and house dust, as well as molecules on cell surfaces of transplanted tissue. When a B cell develops in the bone marrow or a T cell develops in the thymus, the cell synthesizes molecules of a specific protein and builds them into its plasma membrane. These protein molecules are antigen receptors that stick out from the cell's surface. All the antigen receptors on a particular lymphocyte can bind to only one specific type of antigen. Once these receptors are in place, a B cell or T cell can recognize a single specific antigen. One cell may recognize an antigen on the mumps virus, for instance, while another detects an antigen on a tetanus-causing bacterium. The immune system develops a great diversity of B cells and T cells, enough to recognize and bind to just about every possible antigen. A small population of each kind of lymphocyte lies in wait, ready to recognize and respond to a specific antigen. Only a tiny fraction of the lymphocytes will ever be used, but they are all available if needed. It is as if the immune system maintains a huge standing army of soldiers, each intended to recognize one particular kind of invader. The majority of soldiers never encounter their target and remain idle. But when an invader does appear, chances are good that some lymphocytes will recognize and bind to it. After binding to an antigen, a B cell can give rise to short-lived cells that secrete receptor-like molecules that circulate in the blood and lymph. Such a secreted molecule is called an antibody and is specific for that same antigen. Every antibody molecule consists of 4 polypeptide chains joined together to form a Y shape. The tip of each arm of an antibody forms an antigen binding site, a region responsible for recognizing and binding to the antigen, an antigen binding site, and an antigen it binds has a complementary shape, like an enzyme or substrate or a lock and key. A huge variety in the shapes of binding sites of different antibodies enables the immune system to react to any kind of antigen. The main role of an antibody is to mark an antigen by combining with it to form an antigen-antibody complex. Because of their ability to tag specific molecules or cells, antibodies are widely used in laboratory research, in clinical diagnosis, and in the treatment of disease. And in this next slide, we see another table that compares and contrasts the components of the immune system. So please review this slide on your own time. The human digestive system consists of an alimentary canal or gut and several accessory organs. Those organs include the salivary glands, pancreas, liver, and gallbladder. These organs secrete digestive chemicals into the canal via ducts. The human alimentary canal is about 9 meters or 30 feet in length. This long tube fits within a human body because it folds back and forth over itself with many switchbacks. The alimentary canal is divided into specialized digestive organs along its length. These organs include the mouth or the oral cavity, the pharynx, the esophagus, stomach, small intestine, large intestine, which can also be called the colon or the rectum, and the anus. And in this slide, we see a cartoon of the human digestive system. The mouth functions in the ingestion of food. The pharynx connects the mouth to the esophagus, but it also opens to the trachea or windpipe, which leads to the lungs. The esophagus is a muscular tube that connects the pharynx to the stomach. The stomach is a large organ that can store enough food to sustain us for several hours. The small intestine, at a length of about 6 meters or about 20 feet, is the longest part of the alimentary canal. The small intestine is so named because of its relatively small diameter. At only about 1.5 meters in length, the large intestine is shorter than the small intestine, but is called large because it is almost twice as wide in diameter. Where the two organs join, a sphincter controls the passage of what's left of a meal. Bile is a juice produced by the liver and is stored in the gallbladder, and it's secreted through a duct into the duodenum of the small intestine. The last 15 centimeters or 6 inches of the large intestine is called the rectum. The rectum stores feces until they can be eliminated. 2 rectal sphincters, one voluntary and the other involuntary, regulate the opening of the anus. When the voluntary sphincter is relaxed, contractions of the rectum expel the feces. Moving on, skeletal muscles are attached to the skeleton and produce body movement by interacting with the skeleton. Strong, fibrous tissues called tendons connect muscles to the bones. For instance, one end of the triceps muscle is attached by tendons to the bones of the shoulder, and the other end is attached by a tendon to one of the bones of the forearm. The ability to move the forearm in opposite directions requires that 2 muscles work as an antagonistic pair. That is, the 2 muscles must perform opposite tasks. For example, the contraction of the biceps muscle shortens the muscle and pulls up the forearm. The triceps muscle is the biceps antagonist. It contracts and it pulls the forearm down. You need to be aware of 3 types of muscles: smooth muscle, cardiac muscle, and skeletal muscle. Smooth muscle is involuntary, and it is found within the walls of organs and structures such as the esophagus, stomach, intestines, bronchi, uterus, ureters, bladder, and blood vessels. Smooth muscle cells contain only one nucleus and no striations. Cardiac muscle is also involuntary. It is striated in structure and appearance with only one nucleus. Cardiac muscle is found only within the heart. And skeletal muscle. Skeletal muscle is a voluntary muscle and is anchored by tendons to the bone and is used to help move the body around. Skeletal muscle cells are multinucleated and striated. And at the bottom of our slide, we can see cartoons of each of those muscle types. Moving on, skeletal muscles function in the capacity that they support the body, they aid in bone movement, they help maintain a constant temperature throughout the body, they assist with the movement of cardiovascular lymphatic vessels through contractions, and protection of internal organs and contributing to joint stability. Moving on, the skeletal system provides anchoring, support, and protection. Most land animals would sag from their own weight if they had no skeleton to support them. Skeletons also may protect an animal's soft organs. For example, your ribs form a rib cage around your heart and lungs. Humans, like all vertebrates, have an endoskeleton. The human endoskeleton is a combination of cartilage and the 206 bones that make up the skeletal system. The cartilage provides flexibility in certain areas. The human skeleton is organized into two basic units, the axial skeleton and the appendicular skeleton. The axial skeleton supports the axis or trunk of the body and includes the skull, enclosing and protecting the brain, the vertebrate of the spinal column, enclosing the spinal cord, and the ribs. The appendicular skeleton is made up of bones of the limbs, shoulders, and pelvis. The bones of the skeleton are held together at movable joints by strong fibrous tissues called ligaments. Much of the versatility of our skeleton comes from three types of movable joints. Humans have ball and socket joints in the shoulder and the hip. These joints enable us to rotate our arms and legs and move them in several planes. The elbow, for example, is a type of hinge joint that permits movement in a single plane, and a pivot joint enables us to rotate the forearm at the elbow. Moving on, you need to be aware of four types of bones. Long bones that have a longitudinal axis, that's shown in the cartoon on the right. Short bones that have a short longitudinal axis and are more cube-like. Flat bones, which are thin and curved, such as some of the bones of the skull, and irregular bones, which are often found in groups and have a variety of sizes and shapes. And in this slide, we see an overview of the human skeleton. Moving on, the integumentary system consists of the skin, hair, nails, the subcutaneous tissue below the skin, and assorted glands. The skin protects the body from harmful substances and prevents the loss of fluids. Hair on the scalp provides insulation from the cold for the head. The hair of the eyelashes and eyebrows helps keep dust and perspiration out of the eyes, and the hair in our nostrils helps to keep dust out of the nasal cavities. Nails protect the tips of our fingers and toes from mechanical injury. A major function of the subcutaneous tissue is to connect the skin to the underlying tissues such as muscles. There are four types of glands in the integumentary system: sudoriferous or sweat glands, sebaceous glands, ceruminous glands, and mammary glands. This slide shows us a cartoon of the skin, so we can see how the hair shafts protrude through the skin. And when we move on to this next slide, we see a close-up of a hair follicle. And the erector pili, remember we talked about that in a previous chapter. When you get cold, the erector pili muscles contract and form goosebumps. And in this slide, we see a profile view of a mammary gland. Mammary glands are the exocrine glands that in the female mammal produce milk for the young. And remember, only mammals have mammary glands. So let's recap this chapter. At the end of this chapter, you should be able to discuss how different body systems interact with one another. You should also be able to describe the nervous, endocrine, reproductive, and sensory systems, as well as the circulatory, immune, respiratory, and digestive systems. And you should also be able to describe the muscular, skeletal, and integumentary systems.